Hello students, welcome to EPG Pathshala. I am Dr. Janvi Manhas from the Department of Biochemistry, All India Institute of Medical Sciences, New Delhi. Today I am going to talk about Module 16, that is Cell Motility and Cancer. This module is placed in paper 11, Cellular and Molecular Biophysics. Now metastasis is the leading cause of mortality in cancer patients. Despite many years of cancer research, scientists still do not have a definitive explanation as to how cancer cells migrate out of the primary tumor and invade into surrounding and distant organs. These cancer cells enter blood circulation or lymphatic circulation to reach a certain target organ and then leave the circulation to extravasate and initiate a metastatic outgrowth away from the primary tumor. A number of molecular pathways published in literature have contributed to our current understanding of migratory and invasive properties of cancer cells. Migratory cancer cells undergo dramatic molecular and cellular changes to become motile, and this initiates the metastatic cascade. In this video, you can see locomotion of normal motile cells. The most common protrusions by which the cell begins to move and push forward are lamellipodia and filopodia. The number of lamellipodia and filopodia in different cells. In this video, you can see locomotion of normal motile cells. The most common protrusions by which the cell begins to move and push forward are lamellipodia and filopodia. The number of lamellipodia and filopodia is different in different cells. There are cells preferably extended through lamellipodia such as fibroblasts or predominantly due to filopodia such as dendritic cells, and in some cells both structures may be represented equally. These cells are the melanoma cells. Also in this video you can appreciate the motility of cancer cells. Podosomes and invadopodia are specialized structures providing both matrix degradation and cell invasion in cancer cells. Podosomes and invadopodia combine the characteristics of adhesion and protrusive structures. They are not as common as lamellipodia and filopodia. Rather, they are typical for specialized cells and are used mainly for migration of these cells in vivo. Podosomes are typical for osteoclasts, macrophages, some endothelial cells and lymphocytes. Actin cytoskeletal remodeling of cell-cell adhesion and cell matrix adhesions during cell migration and invasion is one of the most critical factors for onset and progression of metastasis. Motility of cells with formation of podosomes and invadopodia is considered as mesenchymal migration. In this module, we summarize the various concepts on the signaling pathways and molecular mechanisms underlying cancer cell motility. The first step in metastasis is migration of cancer cell away from the primary tumor, a process called tumor invasion. To leave the primary tumor, cancer cell has to lose its cell-cell contacts, which is followed by a modification in its cell matrix adhesions. This facilitates migration through the extracellular matrix ECM by either degrading the matrix using secreted enzymes to create their own migration tracks, or by following leader cancer cells or cancer associated fibroblasts that open up migration paths. Using intravital microscopy, it has been shown that cell migration tracks occur naturally in healthy tissues. For example, collagen fibers in the interstitial spaces along blood vessels, between muscle and nerve fibers, etc. These tracks offer paths of least resistance for cancer cell migration. Cancer cell migration along topographical features is non-destructive and does not require tumor microenvironmental remodeling by proteinases. In case of epithelial cancers, differentiated epithelial tumor cells lose their epithelial morphology and migrate to distant sites to form secondary tumors. This temporary and reversible phenomenon is called as EMT, that is epithelial to mesenchymal transition. EMT initiates or augments mesenchymal cancer cell migration. The distant metastasis recapitulates the epithelial morphology of primary tumor, which suggests that upon reaching the secondary metastatic niche, the migratory cancer cells that had undergone EMT reverse these changes by another mesenchymal to epithelial transition, which is known as MET. Cancer cells usually have a primary epithelial phenotype, but sometimes it contains small number of mesenchymal-like cancer stem cells. They may also undergo EMT induced by genetic, autocrine or paracrine factors. You can see in this image that after primary tumor invasion marked as 1, the mesenchymal cells may be better equipped to escape the tumor mass, either by seeding the nearby stroma or vasculature, which may be accompanied by EMT, which is marked as number 2, to acquire mesenchymal features. These cells may migrate past the surgical margins and evade surgical removal, and this gives rise to residual disease. In this image, this phenomena is marked as number 3. Residual disease forms a local recurrence where MET mesenchymal to epithelial transition occurs, and this creates a new secondary malignancy, that is number 4. Transformed mesenchymal forms intravasate into blood vessels. Number 5. Also, a mixture of tumor cell phenotypes including epithelial, hybrid and possibly mesenchymal cells is present in the circulation. This is depicted in number 6. Extravasation from blood vessels takes place, which is again enhanced by mesenchymal attributes. Number 7. After this, the tumor cells may persist as micrometastasis in another tissue marked as number 8. These metastasized cells again undergo MET to establish macrometastasis at distant sites, that is number 9. Metastasized cancer cells undergo both autocrine and paracrine pathways, which modulated the extracellular matrix to promote establishment, survival and growth of the macrometastasis. This is depicted in number 10 and number 11 of this image. Let's understand the role of Cadherin switch in EMT. The invading cells often lose the apical to basal polarity. Epithelial cells with cell-cell adhesion have high levels of E-cadherin and low levels of N-cadherin. However, on mesenchymal transition, these cells gain the ability to migrate and invade, and a key feature is decrease in E-cadherin and an increase in N-cadherin. This is known as the Cadherin switch. As we have discussed in the previous slide that cancer cells have lost E-cadherin, let us understand the pathways causing this loss of E-cadherin. This is a schematic representation of the signaling pathways leading to the repression of E-cadherin gene expression and the upregulation of transcriptional repressors of E-cadherin gene. Let's look at the E-cadherin receptor first. E-cadherin receptor has an intracellular domain marked in green, a transmembrane domain marked in a blue line, an extracellular domain with 5 tandem CAD domains seen as pink rectangles with calcium binding sites in between, which are the small circles. Intracellular domain, that is green, attaches to P120 as well as alpha, beta and gamma catenins. Alpha catenins attach to the actin filaments of stress fibers. This is how E-cadherin is connected to the cytoskeleton stress fibers of the cells. The functionality of cadherins relies upon the formation of 2 identical subunits, which are known as homodimers. The homodimeric cadherin of one cell creates cell-cell adhesion with cadherins present in the membranes of another cell by changing conformation from cis dimers to trans dimers. Therefore, E-cadherin is responsible for cell-to-cell contact. Once the cell-cell adhesion between cadherins present in the cell membranes of two different cells, it forms adherens junction, which can complex with proteins composed of alpha, beta and gamma catenins, binding to the actin cytoskeletal portion of intracellular part of the cadherin molecule. You can also see the IGF-1 receptor marked as a dimer in red. It interacts and phosphorylates E-cadherin and catenins, leading to their subsequent internalization. This is followed by ubiquitinylation by the E3 ligase Hackai and proteasomal degradation shown in the left side of the E-cadherin receptor in this image. In this image, on the right side of the E-cadherin receptor, you can see HGF receptor with c-Met, which assembles a complex consisting of CD44, alpha-4 beta-6 integrin. This together facilitates signal transduction by c-Met. TGF-beta also stimulates SMART-mediated signaling by binding and activating these receptors. All these receptor complexes synergize through downstream effector signaling pathways in inducing the expression of transcriptional repressors such as SNAIL1, SNAIL2, ZEB1, ZEB2 and TWIST. E-cadherin can also be cleaved by proteases to generate a soluble form of the extracellular domain of E-cadherin. Gamma secretase cleavage of E-cadherin results in the formation of carboxy terminal fragment CTF2 that translocates to the nucleus and modulates chiso-mediated transcriptional repression. All these are methods of transcriptional repression of E-cadherin gene, which leads to loss of E-cadherin in cancer cells upon EMT. The loss of E-cadherin affects downstream signaling to cause increased cancer cell motility. By displacement of P120 catenin, which is shown in red in this image from the cytoplasmic domain of E-cadherin, as well as the displacement of IQGAP1 shown in green. IQGAP1 is a scaffold protein for RAS GTPases. This results in the modulation of the activity of Rho family of GTPases. Upon loss of E-cadherin function, cytoplasmic P120 catenin acts as a GDI, which is guanine nucleotide dissociation inhibitor, and represses Rho A, therefore repressing stress fiber formation. P120 also activates RAC1 and CDC42, thereby inducing the formation of lamellipodia and filopodia, as shown in this image. RAC1 and CDC42 in turn repress Rho A activity through the gap 190 Rho gap and inhibit IQGAP1 by binding to it. Together, these activities induce reorganization of the actin cytoskeleton and the migratory behavior seen in tumor cells. The contribution of N-cadherin to cancer cell migration is seen in this image. Schematic depiction of how N-cadherin cell adhesion molecules are upregulated during malignant tumor progression and mediate pro-invasive signaling. N-cadherin has several functions that may contribute to tumor invasion and metastasis. First, cell-cell adhesion to N-cadherin expressing cells of the stroma. So, N-cadherin helps the cancer cell to interact with the stromal cell. The second is binding and activation of FGFR, which is fibroblast growth factor receptor, which is shown in red. This results in FGFR signaling and the promotion of cell survival, migration and invasion. The third thing done by N-cam is cleavage and shedding of the extracellular domain of N-cadherin by ADAM metallopeptidase domain 10. The shredded N-cadherin may neutralize N-cadherin mediated cell-cell adhesion and or stimulate EGFR signaling on neighboring cells. The fourth thing it does is that the cleavage of N-cadherin by gamma secretase results in translocation of the carboxy terminal fragment of N-cadherin to the nucleus. Here the carboxy terminal fragment 2 binds to the CBP, Krebs binding protein, and induces its degradation, thus modulating CBP mediated gene expression. Just like N-cad, N-cam, neural cell adhesion molecule, also contributes to cell migration. N-cam expression is upregulated during EMT and association with FGFR. It associates outside the lipid rafts and in lipid rafts to mediate sustained MAP kinase signaling, which results in focal adhesion assembly and cell migration. Low levels of N-cam forms a complex with FGFR and PLC gamma, leading to the activation of RAF kinase PKC beta 2 and thus to sustained activation of MAP kinase pathway and cell adhesion. Upon loss of E-cadherin function during EMT, N-cam is highly expressed and a subset localizes to lipid rafts where it associates with P59 fin, leading to focal adhesion kinase, that is FAK, phosphorylation, focal adhesion assembly and cell migration. This shows that N-cam mediated signaling pathways are required for cell migration and invasion. Now, cell migration is a dynamic process in which the cell changes shape, produces morphological asymmetry and then translocates the cell body. Cancer cells use similar mechanisms for changing shape, generating force and remodeling the extracellular matrix as normal cells, but lack the physiological stop signal which is achieved by cell-cell contact. In vivo, cancer cells have been observed to migrate individually as single cells, as loosely attached strands or cords of tumor cells, or as well organized collective masses with directional migration. The underlying mechanism in all types of cancer cell migration is the dramatic reorganization of the actin cytoskeleton, forming membrane protrusions like lamellipodia, filopodia and invadopodia, coupled with the cell surface receptor engagement to the microenvironment through which it migrates. Various types of cell migration can be observed in different degrees and combinations. But in general, cancer cells can migrate individually or collectively as multicellular groups. For example, colorectal cancer cells that have lost E-cadherin disseminate as single migrating cells, whereas squamous cell carcinomas invade as collective cell migration type. The nomenclature of interstitial migration modes is based on pattern, that is individual, loosely connected or collective, and further subdivided into typical cell morphology, rounded or spindle shaped. There are three modes of cancer cell migration: single cell migration, multicellular streaming and collective migration. Further, the single cell migration can be amoeboid or mesenchymal. The multicellular streaming can occur as clusters, as a solid strand or as a strand with a lumen. For collective migration, we have amoeboid and mesenchymal type subtypes as well. Let's talk in detail about all the modes of cancer cell migration. The invasive single cell migration results from 5 interdependent molecular steps that change the cell shape, its position and the tissue structure through which it migrates. You can see the 5 different types in this diagram. In step 1, the cytoskeletal polarizes by actin polymerization and forms a leading protrusion at the opposite end of a pre-uropod region, which marks the constitutive rear end of the cell. In step 2, the leading edge protrusion engages with the extracellular substrate, followed by recruitment and adhesion of cell surface receptors that form localized clusters and couple extracellular adhesions to intracellular mechanosignaling and force generation. In step 3, the leading edge moves several micrometer forward, the cell surface proteases become engaged with extracellular scaffold proteins and execute locally controlled proteolysis. This proteolysis modifies the molecular and mechanical tissue properties and allows space for the advancing cell body. In step 4, the small GTPase row activates myosin 2 and contraction mediated by actomyosin generates tension inside the cell. In step 5, this contraction is followed by the gradual turnover of adhesion bonds at the trailing edge, which slides forward while the leading edge protrudes further. So let's talk in detail about the single cell migration, which is of two types, the amoeboid and the mesenchymal. Amoeboid type of single cell migration is seen in leukemias, lymphomas and small cell lung carcinoma. The cells migrate with low adhesion force and adopt a rounded shape. The amoeboid type motility can come in many variants. Cells with short thin protrusions, devoid of blebs and rapidly changing morphology, or slower cells with blebbing morphology and disorganized movements. They can also be found as cells with short protrusions and proteolytic activity moving with slow speeds. In mesenchymal single cell migration, these cells are spindle-shaped cell body and longer protrusions. The protrusions advance rapidly, but the cell rear moves slowly. This results in slow net translocation. The tumors of connective tissue such as soft tissue sarcomas display this type of mesenchymal single cell migration. You can see on the right hand side panel the images of lamellipodia, filopodia as well as blebs. The most common protrusions typical for different cell types are lamellipodia, which are broad, flat protrusions, and filopodia, which are narrow, finger-like, cylindrical protrusions. Also, more specific blebs, that is round membrane structures, invadopodia or podosome. These types of protrusions differ primarily in the structure of their actin cytoskeleton, as well as the dynamics and the molecular mechanisms governing their formation and pulling. As protrusions are the main cell structures providing cell migration, their characteristics determine the effectiveness as well as the character of cell migration. This is a beautiful time-lapse video microscopy of a single cell moving towards a micropipette full of chemoattractant. You can see clearly the amoeboid single cell motility of the cell towards the stimuli. This stimuli in external environment is sensed and reorganization and remodeling of actin stress fibers causes the motility of the cell towards the stimuli. In this case, which is the tip which is actually giving the chemoattractant. This is a video of a fibrosarcoma cancer cell migrating on cell-derived matrix. The membrane protrusions at the front of the cell observed in this movie are typical of mesenchymal migration. This cell was transfected with GFP to visualize the actin cytoskeleton and imaged using a spinning disc confocal microscope. This is an example of single cell mesenchymal migration mode. You can easily appreciate the longer protrusions and spindle-shaped cell body. In multicellular streaming, loose, non-adherent cells move one after another in a single file within the tissue. Individual cells are attracted by chemotactic signals in peripheral connective tissue. It is seen as a swarm-like tissue infiltration of many tumor cells in hematological malignancies and solid tumors. Cells in streams have longer and straighter paths compared to singly migrating cells and have speeds of 1 to 2 micrometer per minute. These cells can display amoeboid or mesenchymal phenotype. Collective cell migration consists of a group of cells which retain cell-cell adhesion and display high correlation of directionality between neighboring cells. Collectively migrating cells form two major zones. Zone A, as seen in the figure, in which a single leader cell or several leader cells generate a proteolytic microtract at the front of the collective group. And zone B, in which the subsequent cells called follower cells then widen this microtract to form a larger macrotract. This is the slowest cancer cell migration mode. The speed ranges from 0.01 to 0.05 micrometer per minute. But faster collective migrations have also been observed. This type of migration is seen in developmental morphogenesis and in most epithelial and mesenchymal tumor invasion. If multiple cells originate from the same location, such as a tumor, the leader cell forms a proteolytic microtract of locally removed ECM barriers, that is zone 1. The following cells then widen or excavate this microtract by mechanical force and proteolysis to form a larger macrotract, which is zone 2. Collective cell migration plays a crucial role in several biological processes, such as embryonic development, wound healing, as well as cancer metastasis. This video shows live cell imaging of collectively migrating Madden-Darby canine kidney MDCK epithelial cells that follow leader cells on a collagen gel to demonstrate the mechanism of collective cell migration. In collective cell migration, protrusion and retraction are coordinated in a supracellular manner, in which cytoskeleton protrusion and contractility are mechanically mediated through cell-cell junctions, allowing the cell group to behave as a large mega single cell. The molecular and physical characteristics of the microenvironment strongly contribute to cancer cell adhesion, migration and invasion. The most abundant and important component of connective tissue is collagen type 1. In healthy tissues, stromal collagen fibers typically appear curly and anisotropic when seen under the microscope. During early cancer progression, the amount of collagen in stroma increases and collagen fibers straighten out. These collagen fibers bundle and orient perpendicularly to the basement membrane to provide tracks for cancer cells to migrate away from the primary tumor. This reorganization of stroma is mediated by stromal cells known as cancer-associated fibroblasts, that is CAFs. CAFs secrete ECM and enzymes that covalently link the collagen fibers. Due to this, the cancer tissue becomes stiffer, while the cancer cells themselves appear to become softer. Cancer tissue is 2 to 10 times stiffer than normal tissue. For example, the breast cancer tissue, which is much stiffer than the normal breast tissue. And this stiffness correlates with the increased risk of metastasis and poor prognosis. Migration modes are also influenced by tumor microenvironment. A more aligned and organized microenvironment promotes collective migration, such as melanoma cells in dermis that migrate along linear tracts of muscle fibers and nerves. In contrast, cancer cells in fatty connective tissue migrate slowly in multicellular groups led by several leader cells. Thus, the diverse and multifactorial involvement of tumor microenvironment in regulating cancer cell migration cannot be overemphasized. Specific alignment of collagen fibers observed using intravital imaging has been termed as tumor-associated collagen signature, which is TACS, TACS, tumor-associated collagen signature, and has been identified as an independent prognosis factor in breast cancer metastasis. This is using all the molecular knowledge to form diagnostic techniques which can directly benefit the patient. Thus, each migration mode is governed by a set of molecular mechanisms, the regulation of which can change the style of migration. Most widely studied examples for alterations of migration modes are the mesenchymal to amoeboid transition or the collective to individual transition. In this image, the thick grey arrows indicate the direction of migration. Cancer cells often lose apical basal polarity and apical cilia and may appear disorganized due to reduction in cell-cell contacts and cytoskeletal reorganization. At this stage, it is called as carcinoma in situ. The cancer cells are still encapsulated by the basement membrane. Due to the crosstalk between tumor cells and stromal cells, the stroma becomes reactive. Now, what is this reactive stroma? This reactive stroma is characterized by an increased presence of immune cells and fibroblasts. These immune cells and fibroblasts help to deposit ECM and reorganize the stromal network. The stromal network fibers are initially loosely organized and appear curly and later increase in density and stiffness. At late stages, the collagen bundles form tracts which are perpendicular to the basement membrane. In invasive tumors, cancer cells perforate the basement membrane or migrate through regions of dysfunctional basement membrane deposition, which allows the tumor cells to invade the stroma and migrate towards the bloodstream. The stromal cells can also enter the tumor, leading to a mixing of cell types and further disorganization of the tissue. Along with pro-tumoral collagen, ECM molecules including collagens, laminins, fibronectin and elastin, cancer cell surface molecules including cadherins, CAMs, proteoglycans and pro-migratory factors like chemotactic soluble factors or ECM bound factors, they all direct tumor cell metastasis and cancer progression. Gene expression profiling and proteomics have revealed abundant sets of soluble factors and ECM proteins which are upregulated in the microenvironment of tumors. This is indicative of complex signaling pathways induced in both tumor as well as stromal cells, and all this supports cancer cell migration. Integrins also mediate the migration of cancer cells along structural components of basement membranes. These integrins engage with collagen type 4, laminins, fibrins, perlecan and versican. In both tumor and stromal cells, multiple protease systems are upregulated including MMPs, ADAMs, cathepsins and other serine proteases. These proteases contribute to tumor invasion and progression through different mechanisms. Cell surface proteases, MMPs and ADAMs cause contact-dependent proteolysis of structural proteins, including fibrillar and non-fibrillar collagens, fibronectin, laminin as well as tenacin and glypican. Proteolytic ECM degradation generates biologically active epitopes of ECM components with adhesion and migration promoting effects. And this remodels the stromal collagen tissue to form gaps and tracks for cancer cell migration. These proteases enzymatically activate other proteases and cell surface receptors, including adhesion and growth factor receptors on both tumor and stromal cells, and regulates the repertoire of available extracellular growth factors by enzymatic activation, inactivation or degradation. Extracellular chemokines, cytokines and growth factors which are released by tumor cells themselves or activated stromal cells engage intracellular pro-migration signaling pathways in tumor cells. Let us understand the plasticity of cell matrix interaction, invasion and tissue remodeling. You can see in panel A of this image that the migrating cells transition from an initial non-destructive dissemination to migration that involves small and large scale tissue remodeling. The pre-existing space available to invading cells governs the type of individual or multicellular invasion. Pericellular proteolysis shown in green in panel A can further help invasion and migration of the cells. Panel B shows epithelial to mesenchymal transition of a stable epithelium after downregulation of cell-cell junctions and facilitated single-cell detachment. You can also see how upregulation of certain molecules can favor EMT or MET in cancer tissue. Panel C and D show that invasion programs display plasticity or adaptability, which includes transition from collective cell migration to individual cell migration and mesenchymal to amoeboid transition. All the key regulators of these transitions are altered in expression or function, and they have been indicated in colored boxes in this image. It's very interesting that a diagnostic cellular arrangement of a cancer cell, a macrophage and an endothelial cell, which is termed as TMEM, Tumor Microenvironment of Metastasis, serves as an independent indicator for metastasis development. To identify microenvironment in human breast cancer samples, Robinson et al. Developed a triple immunostain that simultaneously labels invasive carcinoma cells, macrophages and endothelial cells. This direct apposition of these three cell types is called TMEM, Tumor Microenvironment of Metastasis. In this study, they confirmed that TMEM is present in human breast cancer specimens and can be identified in routine histological sections. It was suggested that this was a useful marker for predicting the development of systemic hematogenous metastasis in breast cancer patients. This is an image of the tuning model of cell migration, and it is an integrated multi-scale model to combine multiple interdependent parameters that impact migration mode. Each parameter can be experimentally tested individually. However, in most cases they are interconnected with others. The format of tuning fork mimics the popular display of a graphic equalizer, which means that adjusting the different ECM or cellular parameters can give rise to a number of migration modes in cancer. The ECM architecture pattern may be dense or loose. Collagen in the ECM may be random or oriented. The format of the tuning model mimics the popular display of a graphic equalizer, which means that adjusting the different ECM or cellular parameters can give rise to a number of migration modes in cancer cells. The ECM architecture pattern may be dense or loose. Collagen in the ECM can be randomly placed or oriented to favor cancer cell migration. The ECM itself can be stiff or soft, which will affect the mobility of cells. The cellular determinants as seen in this image include cell-cell interaction as well as cell matrix interactions. The intracellular proteins which affect the contraction and retraction of stress fibers as well as the number of protrusions made by motile cells are also an important factor for cell motility. Finally, proteolytic enzymes secreted by cells also help to carve a path for cancer cells to move from primary site and metastasize. These are some references for further in-depth reading. You can also mail me if you have more questions. Cancer cell migration and metastasis. Okay. To summarize, cancer cell migration and metastasis is the result of reciprocal crosstalk between tumor cells and the stroma, which includes stromal cells together with the ECM and released factors. In human cancers, tumor and stroma evolve together over months and years. The stromal alterations include cell-derived physicochemical changes of the microenvironment, such as deposition of ECM components, ECM degradation and remodeling, change of ECM stiffness and porosity, and released cytokines and growth factors. Cancer cell phenotype and function contributing to migration consists of changes in the activation, migration and differentiation state of the cell, the metabolic switches in the cell as well as epigenetic changes that may further prompt secondary genomic instability. Appropriate experimental systems recapitulating the tumor and stromal interactions in vivo have to be devised to study the mechanism of cancer cell migration. Consequently, with each cycle of interactive engagement with the stroma, the cancer cell state evolves from its original state. This leads to progression of the tumor and its metastasis. I hope this module has helped you to understand the role and importance of stress fibers in cancer cell motility. I wish you all the best for future learning. Thank you.